Okay, so thank you for the organizer, and it's very nice to have those sessions, and thank you for inviting me, because making the connection with evolution from physics is quite an effort, and it's very, I think, cool to try to bring those communities together, because this is something that's not yet achieved, basically. Well, I'm not going to read you this, and maybe if you arrived earlier, you could read it. I think you can replace mathematician by theoretician in general. This is a wonderful, this is my favorite book. I'm reading it now, and I found this excerpt which I found so much on the point. I mean, you know, it's from 1 century ago, and well, 4 elements became several dozen. We are floating around correlation processes, the dirty dishwasher of processes and formulae. And, well, maybe the main point is we don't know whether it's a thing, a process, a phantom ID, or God knows what. And basically, in my research, it's going on in between those, like, should I try to make a thing, like a molecule? And in the end, I realized that's what society expects from me, to make useful molecules. But I start from processes, so I will show you a bit of all that. So here, what's happening is that Walter is discussing with his wife, Clarisse, and he's very jealous because she had a walk with Ulrich, the main character, and he doesn't know what they told about him. So that's why he's really pissed off at the mathematicians, so don't take it personally. Studying the origin of life. So again, I mean, talking about the field itself, it's a field in the sense that it's a question, and many people of many different disciplines work on this question, but it's not really a scientific field in the sense that there is a community working together. So there is not a clear question, there is not a system of values. I mean, even academically, it's very hard to get positions if you work in this field. I know many people who went to pharma industry in the end. And so now is a new time, I would say, in the sense that it could resemble physics because we can do more experiments. So that's a very, very important aspect of it. And my research is really oriented toward having very close connection between experiments and theory. So I work at ESPCI Paris. It's a school of physics in chemistry, so it's kind of an ideal place in this sense because this research is really at the interface between physics, chemistry, and biology. I would like to start with a little collective thinking, and maybe to warm up, I'd like to ask this question. This is not a scientific question. I mean, you have no way to really answer it. It's more an opinion or a wish. So A, we are alone in the universe. B, there are microbes elsewhere. C, there is intelligent life elsewhere. Well, D, don't answer. Like, come to me after. So who is more in the A category? No one. Oh, you don't. So who is thinking about B? Yes, okay, and C? Yeah, if you answered C, you should answer B, unless you're a creationist. Okay. Well, very well. What about D? D. Yes. So, yeah, I guess all of us have. Or I'm like you. I mean, I'm somewhere here. I really wish to find a life somewhere else. And maybe let me ask you a question. Then, what if you see an article published saying, "Okay, we've solved the origin of life problem"? Sometimes you see that in the news. What type of answer do you expect? What would you like to see in this article, or what do you think is the bottleneck? I mean, it's also a completely open question. Like, yeah, what's for you the most critical thing to demonstrate? What would you like to see demonstrated about the origin of life? But basically, the question is not solved, so, yeah. Okay, yeah, recreate. Yes. Okay. Excellent. Yeah. So in the lab, like experimentally, mix some stuff and observe something that looks like. So this then poses other questions, like what would be your criterion to claim that you have detected life? Like, do you have an opinion about that? Yeah. Yeah. Yeah, the question of the time scale. Okay, so maybe try to elaborate a bit on that, why it's a problem. S- I don't know. So your point basically, but there is an implicit in what you say. You're referring to a reasoning, which is that there is life now, and life appeared a billion years ago. We don't know how long it took, and maybe it took 1 billion years. So what you have in your mind is that, okay, I mix a primitive soup, and I'm going to wait until life appears, and this may take 1 billion years, even if I have exactly the right conditions. That's what you have in mind, yes. So do you say a way around that? Yeah. Yeah. Okay. So your criterion is open-ended evolution, and you understand evolution in a very broad sense. Mm. Yes. Yes. Yeah. Very interesting. Yes. So you're discussing the notion of agentivity, which is discussed in Origin of Life literature. How autonomous is the process is one aspect of it, and I think another aspect of it is you talk, what you refer to in the end is a form of emergent property, and there is a more precise notion, like, is this emergence spontaneous in some sense? Yes, absolutely. So you say, so this echoes to the other questions of the timescales, indeed. So in the end, the first thing that was said is an experiment, but maybe this experiment takes too long, but maybe we can do an experiment to assess probabilities. And if we chain a succession of probabilities that seem plausible and supported both by theory and experiment, maybe we have a chance to give an answer to this question. So yes, I very much agree with that. I mean, that's what we try to do. Other ideas or criteria, like, okay, let's say we publish that, so is everyone happy with this? So some people have your viewpoint and argue that the universe is alive. And would the universe be considered alive even without life as we consider it? You wonder about that? Okay. Yeah, interesting. Yeah. Well, that's a much broader claim, like that there is a sharper uh, line between the two. I mean, you can see the line at from many different angles, but your criterion of open-endedness and out of equilibrium open-endedness could include the evolution of the universe as a form of life. Okay. You had. Yes. So the famous definition of life question. So it's a very vast question that's discussed a lot. I don't remember her name, but there is a young philosopher. She's saying, okay, the strictest definition of life you can have is the pure ontological one, is everything that's in the tree of life. Then, as soon as you try to list properties, you will always find exceptions, but you will also you can imagine systems that you have out of equilibrium systems that you have not observed yet in nature. And I think this is the region where we try to locate concretely.
Yeah, and even the tree of life, right? It's not, as you reach the boundary, there are objects that are not, it's not clear whether you want to call them alive or not alive.
Viruses, yeah, is a classical example. Yes, absolutely. Yeah. So as soon as you put criteria, you find exceptions. So yeah, okay. Yeah. Yes. Okay. Yes. Yeah. Discuss this historical description. Yes. Yeah. Yeah, that would be great. Yeah.
But what you say is related, it's actually related to what I said. Right? Totally. Yeah. The moment you have history stuff,
Yes. Well, that's extremely interesting. But first, to have the viewpoint of physicists also on the question. And we should realize that answering the question is something like up to us. We are reviewers, so we will decide what research will be funded, what articles will be published, and when we decide we have answered the question. So this is also a field where you're directly confronted with that. It's a bit weird in some way. I mean, the epistemological ground is very shaky in some way because there is this subjective aspect. What do you mean by understanding or declaring that we have understood the phenomenon, and this is something that's alive in the community. So what's very important in my sense is that we pose this criteria explicitly and that we are aware that we have this criteria when we have the discussion, because Eric has alluded to the discussion, I will come back to it. There are a lot of fights in the community because people implicitly have a historical criterion or a criterion about the tree of life, and they don't say it and they take it to reject each other's arguments, but this is not really a scientific discussion. But this is really happening a lot. It's shocking when you go to conferences. So the origin of life question. So first, it is a question, and it was not obviously a question before Pasteur, let's say, because there are all these discussions about the spontaneous generation of matter. Like people would see that you have what they consider as non-living matter because they didn't know microbes somewhere, and life, worms or whatever, comes out of it. And so he did experiments where he said he proved that this was not the case. If you seal things and you make sure you kill the microbes, life is not going to appear spontaneously. So I will not say that everyone believed that there was spontaneous generation, obviously, but like it was not clear. And then the broadest question you could say, okay, I have a prebiotic environment. Someone told me once the sky should be red. No, this is wrong. But there is no life, and at some point you have, okay, I would say the simplest form of life, E. coli growing, bacteria growing in colonies. And it looks very simple from here, but inside it's very, very complicated. So we've been through those aspects. It's a different scientific question from, I would say, the one I was used to, in the sense that there is a historical component to it. And some people say, oh, yeah, this is not a scientific question because you will never know exactly what events. But this is not the question we try to answer. It's true, we cannot answer that. And I even had the remark from physicists, you work on that, but it's not a scientific question. But then I ask, okay, I ask you, what was the position and speed of every molecule of gas in this room 5 seconds ago? Is it a scientific question? You have the same problem. It's a problem of inference or reconstitution, and it's a question, can you get enough information, and is your theory good enough? So in this regard, it's not so different. The problem is that the context is quite uncertain. I mean, we have some idea about primitive Earth, and I'm really not the specialist of that, but I mean, there is, for example, this question, where does the water that we see here come from? No one really agrees. I mean, it changes every time there is an isotopic measurement on comets, whether the water came out of the rocks, basically, that were aggregated first, or was transported later after the first Earth dried. This is not very important in the end because there is water in both cases, but if this is a question you ask, I mean, this shows you the level of uncertainty we have about primitive Earth. So the big problem is that the origin of life is not observed rehappening. So you cannot take it as a natural phenomenon in the sense that you are going to observe many appearances of life and try to measure what's happening. So I would say our first task is to actually, ideally, do that, which comes to the first thing that was said by you. And the problem of inference. So Eric has elaborated, I think, the most complete elaboration we can do on that. I mean, how much can you get from observing life about origin of life? But the gap between origin of life and Lucas, so the last universal common ancestor, is extremely big. So there is a region where everything that has happened is maybe erased. So Eric told us about observing biology and extracting constraints, and maybe those constraints are also true in life and in abiotic systems, and they could point to some things, but you cannot look at genomes and say, okay, here is what happened. Maybe the place where it could be done is translation. That's where I believe we can go as deep as we can at the molecular level, but this is a very open question. So because also this is a classification of all the remarks we did somehow. So with a colleague who is a philosopher of science, Christophe Malater, we observed the type of claims in the literature that were made about origin of life. And basically we said, okay, broadly you can classify the difference of criteria according to 3 type of criteria. Are you talking about the historical adequacy of your origin of life scenario? Do you consider that something? Yeah. So, as historical adequacy is, I describe the context of early Earth, and I'm saying something that's compatible with it, and a chain of events that's compatible with the history of the environment, basically. All the measurements of the past. To broaden the criterion, you could apply that to Earth, and maybe you could also have this criterion for another planet, for example. Is there life on Venus? Okay, we know what Venus was, or the moon. Like people believed there was life on the moon before. Yeah, but also the environment that has been there. Well, this is very broad. You can certainly split this axis again into sub-criteria. Is it spontaneous? So natural spontaneity, does it require the intervention of an agent? It has to be discussed every time because, as it was very rightly said, any experiment we will do will be done by a human or a robot that was built by a human. So this chain of causality you have to break it by doing another reasoning, which is a process of naturalization of what you do. You say, okay, it could have happened spontaneously in an environment. My knowledge and my representation of the problem is sufficient to claim that. And there is a criterion of similarity with life as we know it. Because well, you would very much accept, Massimiliano, that I build a form of life that doesn't look like life as we know it at all. Actually, if you elaborate on this criteria, you would maybe accept the universe is alive. So, Eric, you have elaborated as much as possible, I would say, on maybe extracting from the historical adequacy and natural spontaneity, or more similarity with life as we know it. In my case, so the question, the canonical question would be the one respecting all criteria. How did life as we know it spontaneously appear on Earth? And that's the first question you have in mind when you try to answer the original question. So now if I ask, is there life somewhere else? Maybe it's not exactly the question. In my case, my aim is to get rid of all those criteria, try to make, like in this corner, an artificial system that can be considered as alive according to some criteria. It's almost a practical thing because I allow myself to use whatever I find in experimental resources to try to build a lifelike system. But there is, every approach can give to each other, like if you examine nature, you find things that do work. And those things, you could maybe reuse them in the lab to actually build something synthetic. So we all know, I mean, your Massimiliano comes from this branch of science, like this heritage of instabilities and dissipative system. And you know that if you start to bring energy to a system, you can have some structure that appears, and we saw it with Etienne yesterday evening. Okay, is this alive or not? You say, okay, I'm a bit further than not alive, than alive, but could not really say that. So there is this very famous Belousov-Jabotinsky reaction, which was commented a lot also, where you have a cycle and then you have pattern formation. Of course, our brain, I mean, we see something that moves and changes with time, and it's starting to look closer to what you would call a living system, but obviously it's not a living system. But we are kind of reducing the gap. So reducing the gap again, okay, you have the Miller-Urey experiment, which is very famous. You put simple molecules which have CHON, sparkles, heat, whatever, and it's amazing because you kind of form some building blocks of life quite easily like this. But I could buy them.
Using computer animation based on molecular research, we are now able to see how DNA is actually copied in living cells. You are looking at an assembly line.
To have a concrete plan, we must find a way to find even closer implementations. So what's happening inside? Well, I could look at a cell and look at the processes and say, okay, maybe I can take them one after the other. The problem is that if you remove any of metabolism, catabolism, or growth or division, the system dies. So the nature is not telling you in its functioning that there is an order in which those properties could appear one after the other. So that's why there has been a tradition, a strong confrontation. It's more between individuals than anything else, between what's called metabolic first and genetic first. It's kind of the caricature of the debate in Origin of Life. Metabolic first people, I personally don't care at all, say that, okay, there was metabolism and the catalytic metabolism, and then the genetic polymer appeared, and people who say genetic first say, okay, there were the RNA and so on, and after those catalyzed metabolism. Well, when you say it, you realize immediately that it's a bit stupid to think like this. But it's just concretely like the small molecule organic chemistry community is much more inclined to do that because that's what they can do, and molecular biologists and biochemists do that. So it's more community divide. That being said, it poses some clear questions. If we can do that, and Eric showed you the difficulties or potentialities, it's unclear how it would come to evolve. So this is not pointing to a clear order. Genetic first, there is a problem of plausibility of appearance of this polymerase and so on. So those are the two main criticisms from each other. So in the end, we have to construct scenarios, and Eric said it. And a scenario is a proposition for a certain order of events, and then an answer to the origin of life question could be to test every of these steps in a lab time scale and show that, estimate their probability, and then we could believe that the chain of events is a plausible origin of life after comparing it with abiotic conditions, like early Earth. The very stereotypical view is that, but it's hard to imagine, it's very different. You have somehow building blocks that are formed by some chemistry, so is it autocatalytic or not? That's not clear. After, you have some polymerization, oligomerization, and you have more or less random polymers that appear. So what's special about biomolecules is that they fold, and like we are seeing in the talks today, they fold and they acquire some catalytic function. So some of those objects are going to start to change the state of the soup in which they are. And we start this kind of potential for open-endedness somehow. And in a very particular case, there could be a chance that one of these objects is favoring its own appearance, is increasing the probability of its own appearance. So this is a autocatalytic property, or it could be a collective property, which is called collectively autocatalytic sets. And if we start more from the tree of life, we say, okay, there must have been membranes, protocells, and the most obvious thing to copy for evolution is template-based replication. So you have a template, and there is one of those objects that is able to add nucleotide by nucleotide, the complementary chain. So here, at least, we start to have some intermediate concrete criteria of objects to build, process to set up, and maybe imagining how they could chain with each other. And that's what we do in the lab. So all the problem is to be specific, of course, because very often we have to say, like, okay, then something, this is not a problem, evolution starts. Yeah, it's a big problem. So the third session is going to be on compartmentalization indeed, but thank you for the question. You could say, okay, I need a cell because that's what I have. At some point, you have a collective of molecules, and as we saw just before, if you want to have evolution, you must have those lineages in a canonical sense of Darwinian evolution. So it's not just to look like a cell, but it's almost a necessity from the viewpoint of maintaining replicative processes and having a selection at play. So yeah, so it's not clear when it appeared. Maybe it was there here, and it has been scaffolding everything. We don't know. Or maybe it appeared very late. I think that it appeared at least here, but yeah. Well, it's an inference, so yes, there is very strong evidence that it exists. Again, I mean, maybe there is something, it's a bit more complicated in the sense that Eric has been talking about, you could somehow look at genomes and say, Luca was not alone. I mean, that's what is very misleading in this. There are a lot of dead branches here. So Luca has very probably exchanged genetic material with other, so maybe it's, maybe it's not just one cell, I don't know. Don't know. You have an opinion, Eric, on that, maybe? Yes. At least, yeah. But right, it's a good question. It's a statistical object in the end. It's true. Yeah. Came? Sorry. Yes. Yeah, I don't have this slide. There is a nice review of. I will show it tomorrow then. So people trying to estimate the window, and estimating the window of when that could have happened is based on trying to aggregate all possible criteria. So there are criteria about geochemistry, about the formation of planets. Well, within the realm of what I say, it's clear that Lucas is just so much. Well, just the statistical till common thing is, at least for example, the central dogma, ATPases, I think, things like this. They are very complex objects, so obviously they are the result themselves of quite a process of evolution. But then you can try to constrain the window of time of this evolution. Yeah, I will show it then tomorrow.
But the simple answer, tell me if it's correct. Luca is already a very sophisticated.
Yeah, exactly. Yeah, it's very sophisticated, so it could not have been the first. Yeah. All the processes I have shown, they are already completely entangled. So, yeah. Okay. Quite complementary to Eric's talk, I'm going to talk about the RNA world hypothesis. That's what I mostly work on. It's a very convenient, at least experimentally, it's very convenient because you can order RNA, you can synthesize RNA quite easily, that you design. You do experiments, you generally measure their length after reaction on electrophoresis gel. And after, you can do PCR and sequence everything that's in the soup. You have seen Camille poster, maybe, which is very representative of this kind of approach. So what's the claim of the RNA world? So you have the central dogma of biology. And so this book is updated from times to times. And here is a depiction, a very simplified depiction, saying first you just had RNA, and then you had some modification that allowed the codes to become more stable and specialize in information storage, which is DNA. And then you make proteins. Well, this is one view, this is a very caricatural view of the RNA world. So just to remind, the differences between RNA and DNA, this is RNA, ribose, and DNA1. So the RNA is more fragile, so that's why there is this argument. It's prone to hydrolysis, for example. And the bases are not exactly the same. So you have GTAC, and the complementary one are AT and GC. And in RNA, you have U instead of T, and another canonical base pair is GU. So you can, it's more flexible, and you have more possibilities to fold. I really know almost nothing about this, but I'm not going to tell you how the RNA nucleotides formed. But this was the open question somehow, because the Miller-Urey experiment was not showing how ribose form. So people could not really make the NTPs, so the elementary units of RNA. It's only recently that people start to propose like plausible path of synthesis of the RNA nucleotides. So notably, from 2020. So it's a very recent. So some people fight about which one it was, but at least it seems possible. And now there is another question, which is not a simple question: is how do you make oligomers? And personally, I start from oligomers. So there are a lot of open questions before that. Obviously. What changed the game is that we saw RNA was detected very early on, and the genetic code was understood, so it was in 61, I think, in Pasteur. So what was the role of mRNA? And messenger RNA is a messenger, so is a code. In 1982, so Tom Cech and his group discovered that RNA could be catalytic, like proteins are catalysts. It's interesting because he was looking for the spliceosome. So the messenger RNA, it has exons and introns, and some parts are removed to make a functional sequence. That's the one that's translated in cells. And so people try to understand how this process is happening. And there is a spliceosome, which is a mixture between RNA and proteins. And they were looking for a protein enzyme. But the control gels without enzymes, they tested a lot of proteins, were also showing the splicing. So, okay, of course, the student then does like 10 versions of the gels again and again and again. But the control is always... And they discovered that it was the RNA itself that was excising itself from the RNA. So that's how it was discovered that RNA is catalytic. So 1982. And then, so this is basically the scheme. So you have the RNA that kind of falls and guanine attacks. It's a bit complicated, of course, because it has to remove two sides. But this was rationalized. Okay, so RNA carries information and is able of catalysis. So it can do the role of DNA, which carries information, and it can do the role of proteins because it's also a catalyst. So that was the first strong support for the idea of the RNA world. That was interestingly already supposed at the time when DNA and proteins were discovered. DNA replication was understood after the structure of DNA in the 50s. And like Wooser and all those people, they were like, but there is a problem of chicken and egg for the origin of life because we have proteins and DNA. There must be something in between. So they had this very strong intuition. So this was in this direction a first confirmation. So what I just showed are group 1 introns. So introns are those part of the messenger RNA, and group 1 introns is a family of RNAs that self-excise. So when they fold, you can take them alone, and they are catalysts. And this is how it looks. So this is what's called the secondary structure. You have the base pairs that are complementary, are represented like this. And this object folds in 3D. So the different loops, they come together, and they form a folded object like this. You find group 1 introns throughout the tree of life. We have some, so animals. We are animals. In plants, in different... I'll come back to that. So there is another big category of catalytic ribozymes that are much smaller. So it's interesting from the viewpoint of origin. Although they don't perform the same type of catalysis, you end up with ends that's not canonical. But there are small ribozymes. And you see, this is very small, actually. This thing takes an RNA and cuts it. So viruses, they can have cyclic replication, so it makes a chain. And the RNA, they cut the parts of the chain one after the other to separate the new genomes. So those are, this is the name of a virus. And in the cell, there are catalytic RNAs. And they are associated with proteins. So we discussed about the spliceosome, and the ribosome is really, so it's really the monster of the cell. Yesterday, Nassim said, okay, models of proteins become really relevant when you go down to 2 angstroms. So in the years 2000, at Dionat, she managed to do a crystal structure of ribosomes with resolution smaller than 3 angstroms. And those crystal structures allowed to understand that it was not the protein part of the ribosome that was catalytic, but really RNA that was doing all the job, so the proteins scaffolding around. So from this point, it was kind of really hammering the fact that, okay, RNA, at least at some stage, was there. Because if you look at what is translation, it's in the end, catalytic RNA is called a ribozyme. So it's a RNA that takes transfer RNA and messenger RNA and makes proteins. So you can see, okay, in how molecular biology works, there is a form of hierarchy. It's a RNA world that is making the proteins. This does not solve the problem then that when you have DNA, then you need proteins to make them. But you can imagine an RNA world. Well, this is a small summary, but just to show, okay, the catalytic RNA, they come in diverse lengths, and they are a bit smaller than that now. But this gives you an idea of the complexity of the size of the RNA. And also, they do perform a certain variety of reactions, but not a very broad variety. They act a lot on other RNAs.
Because I don't know much of
Yeah.
ribosome, can you elaborate a little bit more on what part is RNA and what part is protein in the ribosome? Because I'm not sure I follow that.
Yeah, help Eric. So the violet part is proteins, and this other part is RNA. So it's a multi-component system first. Yes, so this is one very exciting hypothesis, led mostly by the Lorraine Williams group in Georgia Tech, is that there is a kind of accretive growth of the ribosome. And so the proteins are kind of here to stabilize and coming in the grooves and making the thing better, but RNA is kind of the core of the ribosome. So this is what we see in nature, and then in the 90s, there was a lot of development of directed evolution with all the molecular biology techniques and sequencing and so on. So people started to make completely artificial ribozymes selected from random sequences. This is, I think, one of the first articles of the group of Jack Szostak. So basically, it's always here you're looking for RNA. You design a certain sequence that's random up to in some parts or completely random. And you put this part in the soup and this part, and you design that they hybridize here. So this is a fixed sequence. And you look to try to extract those sequences that are going to catalyze the bond. And that's very smart, because then you have a tag that you can pull out by other biochemistry systems, so you can purify objects. And when you do sequencing, but if you do sequencing up to there, you will see that this has happened. But if the sequencing kind of stops here, this has not happened. So you can associate the genotype, the sequence, with the function which is being catalytic. So this is a revolution of directed evolution. And then with these tools, what Joyce called the biochemist dream, I think, there are some groups, including those of notably Phil Holliger in England and Gerald Joyce in San Diego, starting to try to make completely artificial what we call a self-replicase. So replicase is a RNA that's a polymerase itself that's going to copy other RNA strands into RNA strands. And the dream is to have the self-replicase, which would be an RNA that catalyzes the copy of its own sequence. So if you have that, you have an object that is able to copy itself and to evolve. So this was already shown by Eric, exactly this article in a eutectic phase of ice. Again, it's a serendipitous experiment. They left the sample in the freezer by mistake, and they did the gel, and they said, oh, here is a big bond. Yeah, you should do random experiments. Huh? That's, that's the conclusion. But they found RNA that can catalyze the copy of RNAs longer than itself, which is around 200 nucleotides. The problem is that it's copying, if you look at precisely at the sequence, it's a repeat of 11 mer. So it's only copying very specific sequences, and I asked Oliger directly, I mean, those that do not fold, basically. But then it cannot copy itself, because to be active, it has to be folded. So you are not going to solve this problem by temperature. It's not like strand separation, where you can imagine a phase of the reaction where you make the strand. This is one problem. You have to separate it to restart again. Here, it's at the same moment you are trying to copy the same structure that is folded. So that's what people are doing now. The biochemists, they try to make those objects, but that really handle the RNA tightly to process through the folds. So that's the current problem in the field. So obviously, people are progressing. Maybe at some point we have such an object. So currently, this is the most recent article of the Joyce group. There are replicases made of RNA that can copy other catalytic RNAs, and you can observe evolution. But they cannot copy themselves. So it's not an autonomous process. So I'm going, there is, if you make these objects, there is a next problem that awaits for you, which is the problem of the error threshold. I'm going to derive it on the board. Up. Anne Florence has shown you this graph that I will show you too, and maybe some of you have noticed that the length of the genome and the error rates were inversely correlated. It's another version of this graph that you have seen in Anne Florence's presentation. And there is a good reason for that. Which was discovered by Manfred Eigen. So interestingly, it's a chemist who got the Nobel Prize, I think, for stop flow chemistry, and he was thinking about this problem of replication, original 5, and he found this relationship, which is quite important for biology. So you have a relationship between the length of a genome and the error rate it can have. So there is a very simple manner to derive this in a very simplified model. The original article of Eigen considers a list of species, and I'm only going to consider 2 species. The species that replicates itself, and one erroneous species that we'll call a parasite, which is a species that appears when the copy makes an error. So the setup is that you have your replicator. And here you have the parasite. And you have a self-reproduction rate alpha, an error rate mu, and a self-reproduction rate gamma. The question is who wins in this dynamical system and what happens depending. So either this one wins, or this one wins. Or I can give you the result very intuitively. If this one wins, it survives, and with a certain rate, it's going to give this one, which also survives. If this one wins, but this one dies, because there is irreversibility here. So what does it give in numbers? Well, I'm going to rename, okay, I can, it's very elementary, but you have a dynamical system where here I call M and Y, and I have a matrix A, C, B, where I identify A with alpha, so it's just a notation. B would be gamma, and C is mu minus alpha. Alpha minus mu. No, sorry. So A, the self-reproduction is alpha minus mu. C is mu. No. B is gamma, and C is mu. I just take the exponential of this matrix, and what's going to happen is that I have a dominant mode. So you know it's a upper diagonal, lower diagonal matrix, so the eigenvalues are A and B. And depending on whether A or B is larger, I'm going to be dominated by the eigenvector. The composition of the system will be the eigenvector corresponding to the largest eigenvalue. So there are two cases. If B is larger than A, then the composition is going to look like this. So this is a case where the parasite wins. And what's important is that the eigenvector has a 0 and a 1. You can, it's immediate. And if on the contrary, the other wins, then my eigenvector is going to grow like this. Yeah.
The reaction is irreversible because there are many parts.
Yes. Yeah. Absolutely. Yeah. Absolutely. So I will show after the derivation has been done in the case where you have a form of reversibility, which improves the error threshold. So it's less bad to if sometimes you can revert. But in practice, there are two aspects to it. Sorry. Like either it reverts, but this is very, very rare, as you said, or there is a neutral space. So there are some mutations that are not causing problems. Then you can preserve activity also longer. This has been calculated. Yeah. So in the end, M survives. Is equivalent to saying that if I replace the values, alpha minus mu is larger than gamma. I call this star. The question is, okay, can I estimate mu as a function of the length of the molecule? Because this is a process of copying. So each time I add something, there is a certain probability that I make an error. So now we are interested in considering, as in the final relationship, epsilon the error per nucleotide. L, the length of M. So the probability of a successful copy, so error-free, is this. I never make an error L times. And this is by definition this value, by definition of the graph there. So it's almost done. Right? The final, you see here that you have a relationship between the error, the length, and some parameters. I combine this with this. So that's what I'm going to do. So all the ingredients are here to have the error threshold. So basically, I take the log. So now I take my relation star. Which becomes equivalent to this. Yeah. Yes. Yeah. Exactly. Yeah, and the error threshold comes from taking the log of this. So when epsilon is small, the log of 1 minus epsilon is approximately equal to minus epsilon. So you end up with the key relation. If you rearrange everything, which is that you cannot maintain a genome longer than L being defined by the ratio between log of alpha over gamma. So this is called is a selection coefficient in this context, is how much better does the good sequence grow over the bad sequence. And I divide by the error rate. Yes? Here it's defined as S, but I would say in a context like the one of Anne Florence, it would be called 1 plus S. So the original derivation is considering a list of parasites, an arbitrary, which makes the calculation look more complicated, but in the end it's the same calculation. You also, in the original paper, have minus a certain dilution term, a total dilution, which is equivalent to saying that the total concentration is constant. So this is a theoretical convenience, but we see here it's not very, yeah, it doesn't matter too much. It's just who wins or not. I will go back to the slides. Well, what's really amazing is that this relationship is followed in nature, apparently. So you have small genomes of RNA viruses. They are very prone to errors, and the very large genomes from us, we 3 billion base pairs. We have specific error correction mechanisms to be able to maintain our genome. Okay. Amazingly, there was a first, I think it's the first directed evolution experiment, some artificial experiment, the experiment of Spiegelman, which is extremely famous to molecular biologists. Before there was PCR. People were amplifying RNA with the Q-beta replicase. It's a protein from a phage, I think, which copies RNA into RNA. So this kind of object does exist in nature, but it is a protein. It is not an RNA. So it has a unit that hijacks the unit of the usual polymerase of the host, and then is able to copy RNA into RNA. So he did serial transfer experiments, as we have seen before, but amplifying the RNA of the virus artificially outside of a selection context. And what he sees is that the infectivity at some point completely drops and it loses its activity. So that's how he detected that there was an accumulation of mutation and loss of the RNA. And you end up with a parasite. So it's a direct illustration of this experiment without selection. A direct illustration of the threshold without selection. Well, just to re-elaborate a little bit on that, now what does it mean in terms of origin of life? Well, this is fidelity, so epsilon equals 1 minus q. It's equivalent. Problem, possibly I will come back to that, is that, okay, you make a replicase, but if it's too much error prone, then it's going to collapse for dynamical reason. So not only you need RNA replicase, but you need it to be fidel. The problem is that to be more fidel, you need to be longer because it's a more elaborate catalytic property. So it's not known if there is a replicase that is long enough to be fidel enough for its own length. That's what's called Huygens paradox. The problem is that so far, and this is really the favorite argument of creationists, like any replicase you can find that's really not so good in the end, is something like 200 nucleotides, and the combinatorics of it is huge, like it's 10 to the 120. Alexandra said she doesn't want to talk about these numbers, but I do. Like the number of atoms in the universe is 10 to the 80, 81. Okay, it's this is not realistic right now. Huygens proposed a fix to this problem. He said, okay, maybe there is a cooperative process. He called that hypercycle, and it introduced a lot of confusion in the mind of people. But people really like this idea first. It's the idea that you have replication system that make a product that is going to help another replication system, and the mutual help between them would favor the survival of the overall system. I want to highlight the fact that you need each subsystem to be itself a replication system, and that's not specified in the model very clearly, so this model is not very specific. Okay, this was fashionable until the students of those guys, years after years, they studied it, and it's very prone to instability. So this is, even if it's complicated and it's not helping so much. So a bit related to the question of Juan, there is this notion of relaxed error threshold if you include the fact that some mutations are neutral. So you take a RNA, maybe, we'll see that in detail, but if you exchange those two bases for two compatible bases, you are not going to change too much the fold and the function. So there are mutations that are neutral in the sense that they don't change the function. You can improve the threshold. Okay. The problem, the derivation I have done, is that it's a linear one. But when we talk about the self-replicates, it's not linear because you are talking about something that is its own substrate. So you start to have some linearities kick in, and it's absolutely horrible. Like it's even more catastrophic than the error threshold itself for the self-replicates, which was shown in this article. And for having done experiment that I will show you, like Spiegelman, what you observe is that you have a shortening of the RNA. That's the first thing that happens, not mutations. Then the selection coefficient here, because the time is approximate to replicate is approximately your length. This is smaller than 1, the ratio, which means that the log of something smaller than 1, the length is 0. So as soon as if your replication stops somewhere and then you have a parasite that's too small, you're just immediately dead. So I think in practice, no one says it, no one discusses it in the literature. It's seen nowhere, but I tell you, this is the main problem. And then something that's more hopeful, and I will elaborate in the last talk, is that you have compartmentalized selection, which is somehow elaborating also on what Anne Florence has shown. The very dumb version of it would be to say, okay, I compartmentalized, and when I have too few good guys, or if I have too many parasites, I kill the compartment. So it's a way to manually I remove when information diverges too much. In an extreme case, if I select them one by one, of course, it's going to work. I only keep those that are good. But it works even if you have small collectives. So this was called the stochastic corrector model. You have two levels of selection. You have the replication dynamics inside the compartment, and you have the compartment survival that depends on the probability, on the fraction of parasites in the compartment. So this is coming back to the question of when the proto cells appeared. Yeah, maybe they had to appear very early if you want to have a sustainable replication process. Maybe you need proto cells as soon as you have a replication phenomena. Well, I'll jump that. Okay, so what we work on in the lab is the idea that maybe before you did not have suddenly a self-replicase that popped out, like with this one super magical sequence from nowhere, but you could have had a process of evolution that's more gradual. This is not so much studied. I mean, I think the idea is coming more and more. But why I like it, and also why it's a bit difficult, is that you need to combine theory and experiments because you are talking about collective self-organization. So you must set up systems that are sustainable but are made of several components. The literature of biochemistry is starting to hint at those ideas. So the same group I showed the replicase of Holliger, they proposed, okay, maybe this object, it's assembled from pieces. And I have an object that is going to stick those pieces together, and then this final replicase is going to copy each of the smaller parts. So they gave a proof of concept that this is kind of possible. And the same group is working on maybe, okay, it's very difficult to add nucleotide by nucleotide, but maybe I could make my object from bigger chunks, precisely from triplets. Because then I improve the penetrance to folds, which was a problem, and I increase the specificity also of my building blocks. Yeah. Okay. Yeah, I don't know. Yeah, I don't know. I'm not updated on that part indeed. Yeah, no, I don't know. Yeah, okay. That there is a correspondence between the codon length and this, the basic unit of 3 nucleotides. Yeah. Well, at least they work on it and they can copy folded subparts of the molecule using this trick. But this is not yet also working as a self-replicase. This is ongoing work. Yeah.
At this point, if you are copying the blocks, why do you need a single self-replicase? Couldn't it be our network of replicases of one copying only some part?
Absolutely, yes. I think there is an experimental reality that you cannot really do what you say because you have to study one object. Yeah, this is just not studied, but why not? I think to motivate people, you will need to make a model that shows that this is better. And then maybe, yeah. But yeah, I mean, this is not wrong at all. It's a line of thinking that's worth exploring. There is no theory attached to it. Those are experiments going on. No one is doing a model of this. No, for you, if you look for a problem. So if I push that to the complete extreme, so we all have the gut feeling that, yeah, it was not this super magical object that appeared. There is a form of collective self-organization. And this idea was, I think, the first time made clear enough by Stuart Kauffman, notably in this article, where he looked at binary polymers, which are A, B, so this is monomers, then you have polymer AB, BB, etc. And looking at fragmentation, condensation reactions. So you take 2 of these polymers, you make a bigger polymer, or you cut it. And he said, okay, I have a reaction network. I take the vocabulary of my binary polymers, or it could be proteins, and I consider all possible fragmentation and condensation reactions. And there is a very simple enumerative argument, which is you want to estimate the ratio between the number of species and the number of reactions. Because it was known at the time, I mean, all the percolation phenomena were understood well enough, and also the question, what is the probability to have a cycle in a random graph? Typically, it's a threshold on this quantity. If it's larger than 1, typically you would have a cycle somewhere. So you just enumerate for a fixed total length. So you look at all the polymers up to length m, and you enumerate the number of species divided by the number of reactions. There is a detailed calculation, but in the end, you find that this ratio is equal to the maximum scales with the maximum length of the objects you consider. But hence, at some point, whatever the probability P of catalysis that you suppose, so you take all reactions, you say, okay, it's going to be exist or not with the probability P. I multiply m by P, and if m is large enough, Pm is going to be larger than 1, and I'm sure to have a cycle. That was the argument of Kauffman, basically saying, if I have a diverse enough chemistry, the probability to have a cycle, so a closed loop of reaction of something that in the end is going to make itself back, is certain. Well, this has never been verified experimentally, because you would say, okay, now I claim that, I just take random polymers and I'm going to have an autocatalytic cycle. No one dared really studying this problem, in fact. But now, I mean, we study it, and some people started before me, obviously, but there is one thing I would like to remark is that as any percolation phenomenon, you know it can be very abrupt. So one potential problem is that if you have one cycle, it occurs that everyone is connected within the same very big entity. It's a big, strongly connected network, and that's also bad for self-replication because it's a mess. So you do not have this kind of phase where you have a lot of different isolated self-reproducing cycle that exists if you consider a random chemistry like this. So after, there were some work saying, okay, but your parametrization of the probability to be a catalyst is wrong. You cannot just take reaction and say, oh, this is binomially. So those are lines of research. It's not very clear. Well, I have a hypothesis, but I will, which is very similar to what Eric has shown. I will discuss it at the end tomorrow. So we work. So nice. The group of Nice Lehman developed a system that the system we use, which is based on the group 1 intron, is the one I showed you, like that Tom Check has discovered in the 80s. They are autocatalytic systems made of RNA. So here you have fragments that come together by base pairing. Complementarity. So this is a complex, and you see you have dangling ends that are not covalently bound, so it's not a very stable object. But if you have a fully covalent object, which is, it's able to catalyze the covalent bonds, so to attach the parts of the RNA together. And so you have then 2 copies. And then each of the 2 copies can again make copies, and you have an exponential amplification of the process. So this is topologically very different from template-based replication, because here you have a complex, and then it's attached, and things are not separated. It's a new catalyst. So it resembles much more it's kind of an illustration of Kaufman ID. So this object does exist. It does reproduce, and we use it in the lab. Now the difficulty is that it's not clear how you would have evolution from that, and that's all the purpose of our research. So there are 2 systems like this. The one I just showed you. So again, you have fragments. They come together in a complex, and once it's covalent, it can further make the covalent bonds between other complexes. And you have the Joyce ligase, so-called Joyce ligase, where you have pieces that come on a full-length object. This object is able to catalyze this bond. And then you have the reciprocal catalyst, which is going to attach unligated small pieces. So those are the 2 self-reproducing RNA that we know, and they work with autocatalysis, not template-based replication. So we want evolution. We have not said, I think, in the talks so far, what was Darwinian evolution. So I have to say it. Well, this is a picture of Darwin, you know. And okay, so Darwinian evolution is synonymous of evolution by natural selection. There are a lot of articles commenting how it should be defined, but a very common way to say it is that you have 3 properties: variation, reproduction with heredity, and selection. This is a Wikipedia image, but you have different types, and there is selection in the sense that they would or not survive. Some types are able to give the same type, more or less, or other types. So you have a tree in this picture, and with different states that correspond to different survival. And I mean, when people understood how evolution occurs in biology, this was such a strong revelation. I mean, it's a complete epiphany that we cannot dissociate what we see in biology from Darwinian evolution now. But at the time of Darwin, I had no idea. And what I want to say in this way is that you can consider an evolutionary process independently of the idea of template-based replication. Template-based replication is a particular implementation that's extremely powerful, but maybe there are other ways to do Darwinian evolution. So for example, the black thing is the group 1 intron I showed you. that's able to do autocatalytic self-reproduction. We've not published it yet, but what we intend to do in this kind of review article is to list all the type of reaction schemes it can perform. And here in the box, there is a kind of a vocabulary of operations it can do between fragments. So you can recombine objects. So you can take I and put it outside. That's what it does naturally. You can take a piece and attach it to the next one. You can remove a piece. So you have kind of a series of operations, a vocabulary of sequence transformation. And I think a question is, can you use this vocabulary and build a system that's Darwinian? To concretely relate to Darwinian evolution, if you can do recombination, you can do variation. That's happening in our cells. So we have self-reproduction, we have variation. The big piece that's missing is heredity. So that's Camille's poster. That's what we are trying to achieve in RNA reaction networks. To have pieces that can be amplified reciprocally, like in the ones I have shown, but catalyzed by this object. So then once we will have this piece, I think we could build reaction networks which combine the Darwinian properties. Yeah, but I'm going to more or less stop here.